So I have a few story and a few methods that I want to discuss today, but I'm not sure I will have time to discuss them all. So maybe at some point I will ask you, what do you prefer in case we run out of time? But as Lionel said, like the main topic of this of my work and the field within I'm working is about super resolution microscopy. Let me see. And I don't know how many of you has been using a super resolution microscope, but probably you know that is like this sort of new field in microscopy compared to conventional microscopy. So it has been like really pushed and developed in the last decades that enable us to make actually dive into the spatial resolution and get information that are typically beyond the diffraction limit of light. So approximately, like if you use a confocal microscope, focus light, visible light, you can get information around, 200 nanometers. And you know that this is still, order of magnitude higher than the size of your tiny molecules. So with super resolution and with different technique, you can actually start getting information at 100 nanometers, 50 nanometers, and now with the recent advance, even a few nanometers. So that means in terms of fluorescence imaging that, you move from recording this type of images to this type of images. Here, the resolution is around 40, 50 nanometers. And the system that you are looking at are neuronal cells that we like to study in the lab quite a lot because they have synapses, which are quite tiny compartment. This is, you see only one side of the synapse, the postsynaptic side, call it also dendritic spine, because are like the spine in the rose as a plant. And then you see here two different molecules, one labeled in magenta and blue. So you can already appreciate that by the increased spatial resolution, you can start asking new questions, maybe better question. I'm not saying you are solving the question, but at least you can start asking, does this protein cluster? How big are these clusters? How many molecules are there? And do they really colocalize? Before, you could have said, oh, these two molecules are both in the spine head, but now are they really like colocalizing and in which way they are structuring? So, and this is just one of the questions that you can ask. My personal dream is to contribute with methodology to get this information as more dynamic as possible. So I really want to study how dynamic overall happen at the small scale. So you haven't heard before about super resolution, and I know that Fernando was supposed to give you a very nice introduction about the method. I will not have time to sort of pinpoint at the different technique, but I will describe in really details the technique that we are pushing in the lab that are instead resolved. But for you to know, there are like two main different concepts in super resolution, the one based on single molecule detection, and the one based on, we call it ensemble. So the trick in one case is that you want to have always one single molecule per diffraction limited spot. So you trick your sample so that you have a sparse labeling. In the other case, and that can be, with photoactivation or so on. In the other case, that is what we are going to talk about today, you need to have an illumination that is modulated in space. So you need to have a pattern of light. It can be a donut, can be multi-lines, and so on. But you do need this modulation. And then you also need to have ways to make the molecule visible or not. And that we call it sort of molecular transition that are reversible. And I'm going to mention some. Some happen through electronic state, other to conformational change in the molecule. All right, so a few words about what we do in the lab. So if you want to develop methods in general in super resolution, sometimes when I say I'm in microscopy, we do optical engineering. And for a long time, that was enough to create new microscope. But then over the years, what became more and more important is to think about the probe. So now this is part of your microscope. So what you decide, so how you decide to label your sample, but not only the labeling, but actually how the physical chemistry of the probe behave. And in our lab, we really give a lot of importance to that. We think that this is, exciting. And if you understand new kinetics, new mechanism here, you can really drive the methods much forward. And then, of course, you need still the optical engineering. You need to develop software in order to, in a way, get the best out of this transition. But they come together. They are not disentangled anymore. And then eventually, we like to apply our new methodology to challenging system. In our case, we already choose neurons that are challenging because they are primary cell. They're not that easy to treat compared to other systems. And also we like to look at them in 3D and dynamically, which pose additional challenges. So like when you develop new method, there are a few parameters that you should keep in mind. And they sometimes don't, improve in the same direction. So for some people, this is named the triangle of frustration, the microscopist. And that means that, when you want to increase the spatial resolution, you get slower. Why? Because you need to sample more and eventually to add additional light for enabling this transition. And also, so that will bring you then far away also from the light exposure, right? So you become more phototoxic. So, and there are triangles, and then that we call a trade-off, right? Every technique has its own trade-off. And so if you want to get to live cell, you have to take even more care of this, because you still want to resolve as good as you can your sample to get the high spatial resolution. But you also don't want to fry the cell, right? Because then, what you are seeing, the dynamic that you are seeing are just light-induced photo damage. So you want to avoid that, and you have to keep an eye and do the right control to see that you are not affecting the cell, or at least not the process that you are studying. And then there is the speed part, right? That what I said, like if you want, most of the microscopy are familiar with the Shannon-Nyquist principle. So for example, if you want to have a confocal microscope, you want to find the scanning step, maybe it's enough 70, 100 nanometers. But if you want to get resolution that are 50 nanometer, then you have to sample every 20, and then things get slower because you need more pixel, you need more light. And so this is also a bit of a nightmare. So you have to think, and that's what we think a lot about, ways to, make the triangle smaller. So exploring different trade-off. And that let us, me and my lab, study for a very long time and developing a technique named Resolved. That means a reversible optical fluorescent transition. And is based on the photo switching of this fluorescent protein or probes that change conformation. I will talk about that. But the interesting part is that when you switch this probe between these two states, the states are long-lived. So that allow you to move from using, for example, very high peak intensity to different way, to like lower peak intensity and maybe lower even light dose. So to deliver the photon in a very different way. So in this way, we can be gentler, because one of the things is that photo damage is a non-linear process. So it really matters how you deliver your photon. And this is just an example that I always look sort of to motivate me, my lab mates, that is so showing between the sort of known super resolution technique, like how much light do you need to record a single frame. And then you see like, STAD, PALM, SMLM are like sort of the most famous, but still in different ways. So STAD use high peak power, very short time, and single molecule you use sort of lower intensity, but for longer time. They still overall need quite a lot of light dose. While with our reversible switching, and this you see PS, Resolve, and Mona Lisa, we can decrease it simply because this protein can switch with less light. And same for speed. But this for another reason, because you need less light dose, you can actually enable yourself to build different microscope and sort of going through parallelization, like confocal versus spinning disk, and then to get also faster. So what is Resolved? So Resolved, in a sense, is, as I said, is an ensemble technique, also named coordinate target switching type of microscopy technique. And the way it works, practically, or in terms of optics, is very similar to STAD microscopy. So that means that you need to focus your light. So typically you use a high numerical aperture objective, and then you have your double cone of illumination. And this is what happened in confocal microscopy. But then you know that your volume will still be, 200 nanometers versus 500 nanometers axially, which means that, you cannot distinguish distances that are below 200 nanometers. So if you make a line profile, it will look like this. But then instead, what you do, instead microscopy, you add an additional pattern that is a donut and look like a toroid in 3D with a central zero. So this means that only the molecules that are here and here, or if you want to see in 2D, here in the periphery of the focal spot, are the ones that see the red photons, right? So in the molecule that see the red photons are the ones that get depleted. So they are emitting, but not at the light that we detect. So the result is that the ones that interact with their photon are off. So the only one truly located in the zero at the center are able to emit. And those are located then in a very nano-sized volume. And this is exactly where you get and generate the high spatial resolution information. So this is the concept, like basically depleting the ability of the molecule to emit fluorescence and to let only the few one in the center still able to behave like a normal conventional fluorescent molecule. So results instead on this side are the very same. We do need the light pattern, we do need a zero, and we do need to switch off molecule. And then we do need to do that reversibly, because then you move to the next point in the sample and you have to do it again. So the very same molecule that were off need to be on again. So you need the reversibility, which is not the case, for example, for single molecule type of technique. Okay, so then you're wondering, are you just reselling me STAD, with just a different name, Resolved? What is about? No. So what is different is the way we switch off the molecule. Like in STAD microscopy, the donut, what it does, it acts here on the electronic state, and then it does stimulated emission. So it depletes. So it forces the molecule to emit a photon that is the same of the STAD beam, basically, or very similar. So basically it's red shifted. And this is a nice transition. It's very fast, it's immediate, but you have to deplete before the molecule emit fluorescence, which means that to be efficient, you need to use intensity that are in the order of megawatt and gigawatt on centimeter square. Basically tens or hundred of milliwatt in the backfocal plane of average power. In Resolved, instead, you use long-lived dark states, which means that you have a molecule that behave in a certain way, like a GFP, for example. And then this molecule is also able to shift the absorption and emission spectra eventually upon light. And it can do that reversibly. There are chemical parts like protonation, deprotonation, cis-trans isomerization that enable this switch. So it's a conformational change. It's not only one electronic state, right? And more interesting is that this state can be long-lived. So when you kick the molecule in the off state, it can stay there. So you don't have to compete with fluorescence emission, which means that then you can use much lower intensity. You don't need to hurry up and give as much photon as possible to be efficient, but you can deliver them in longer time. And this ends up being more gentle with the sample. But you do need special probes. That, to be honest, while STAD microscopy, like stimulated emission, is a characteristic that is more general. Still, there are probes that are better than others, typically the brightest, because you also induce more photobleaching, but it is more general. While in our case, we have to use these fantastic tools that are called the reversible switchable protein, and they are, enabling all this type of technique. So the most famous one is this reversible switchable enhanced green fluorescent protein, or DROMPA, that were the very first photo switcher and still one of the best. How do they behave? Typically, they are in the green on state. Then you excite them with blue light and they emit green photons, and they do that for a certain amount of time until they just get kicked in this off state with a chromophore change. And when they are in this state, if you shine blue light, they don't do anything. So then in this state, you can shine UV light and you bring them back, like UVVs, 4 or 5 nanometer. And you can do that really 1,000 times. If you do with the right light dose, they have very low fatigue, so they can switch many, many cycles. And then now we start really teaming up a lot with protein engineers, and we get more and more protein also in the red. They're quite interesting. They start to get as fast as the green. We still have to work with the brightness because we know that shifting in the red is always an issue. You get less brightness, but maybe more excitation coefficient. So this is another way to see these kinetics in a bit more quantitative way. So these all are switching protein. Maybe you might have heard Skyline is also used for SOFI or SURF, GMARS, RSGFP, DROMPA. So it's basically like that. If you set up an experiment where you pulse blue light, they just switch off. And then they can do that with different kinetics, so they make them faster or slower. And then in this one, we are basically shining blue light, and the 405 blue light and 405, we are checking for how long the fluorescence stay. And then that's what we call fatigue. Like we like this one because, you know, it can cycle many times. And usually the faster, like DROMPA, RSGFP, they are really great here. They give you less photon per times, but then they cycle more times. And here, they cycle a bit less. And these are all parameters that are very important for super resolution, like how fast you switch, because, you know, you don't want your dwell time to be like last forever. And also how precise you can switch, because that determines how good you are switching off the molecule in the periphery of the focal spot. And here is just an example again of how your resolution improves basically, or how your confinement of the signal improve. So basically you see here, that would be your diffraction limited spot. And by applying the donuts, all the black dots are the protein that you were switching in the off state because you were shining blue light. And the one at the center are still the one that haven't seen the blue photon, so they are still in this on state. And then you collect the fluorescence of those. And again, the difference with STAD is that this formula is the same that you would use for STAD, but the IS is the saturation intensity. So the intensity that you need to switch off 50% of the protein. And that's very different because this depends on the lifetime of the state. So longer the lifetime, less intensity you need. And so, you know, if you work with STAD or Resolve, you find a lot of these saturation curves because it tells you how basically good you are confining your fluorescence. Okay, so then you want to build a microscope, a Resolve microscope. The first implementation we did was still a point scanning microscope. So very similar to confocal microscopy, but instead what we do, so we first activate the protein, and then in the very same point in space, but a different time, we shine the donut in order to confine, and then we shine another Gaussian in order to read out the few one left in the on state. So we pulse these three pattern pixel by pixel, and then we scan either the sample or the light across the sample. And these are the very first results about Resolve that were really encouraging, because basically they were telling us, yeah, you can do live cell super resolution, right? You can express your protein, your RSGFP2 inside your living cell, and then you can track basically dynamic and over time, and the resolution it is comparable to what you get with STAD and YFP, for example. But the good news was that we needed just this light to build a system, which are really a few microwatt on the backfocal plane compared to what I said, tens of milliwatt or 100 of milliwatt that you would use in STAD microscopy. So much less intensity, which is good for phototoxicity, but is also good for technical implementation and thinking about different optical scheme. But as excited as I was at that time about the system, I also realized that, every system has its pitfalls. And in that system, we were still relatively slow. So if you notice, like to get decently fast, so like second frame rate, we had to reduce a lot the field of view. And the reason is that, you need to switch completely the molecule pixel by pixel, and this will still take at least half a millisecond, so quite a longer pixel dwell time compared, for example, to STAD, which you can use a few tenths of microseconds. So then how do we solve this issue? Then we thought, okay, we have to think in a different way, because if we want to record and take image of whole cell, and now everyone want to do single cell, whole cell imaging, we would need to require so many pixels, and that would increase the recording time up to hours. It's not really lifestyle compatible, right? It's impossible. So we can only work in tiny field of view, basically. So how do we solve this issue? Well, we think about parallelization. Like microscopy in the past has been doing that. Instead of using one beam of light, why don't you use many of them? Then you can reach and jump and very different trade-off. And that's what we want to do. It's a little bit of inspired by what they did with spinning disk, right? Instead of using one dot, you have many illuminating at the same time. So the large field of view come almost for free. So we can do the same with super resolution. We just need to be much better in designing our pattern illumination. So basically what I'm saying is that instead of using one donut and scan the light across the sample, which would take this time, that depends on how many pixels you have, so your sampling frequency, we can design many donuts or many holes, and then just scan one periodicity. And you see that you lose the dependence on the size of the field of view, because that depends just on how many donuts you create. So the rest come for free, right? You just scan one thing and the rest repeat themselves. So that's what means parallelization. So the way we do it, we use a microlens array for switching on. We use interference patterns that are very similar to the one that you have in structural illumination, but I will come back to that to switch them off. And then again, microlenses to read them out. And typically we like to decouple the periodicity, so to have for switching off, like instead of the donut, like this very fine sinusoidal pattern, so that we can confine with very little light. And to read out, we like to still have a little bit of higher periodicity so that we minimize lateral, but especially axial crosstalk, so we are better in sectioning. And this is the latest pattern that we created to achieve super resolution, not only in 2D on the focal plane, but also in 3D. So Andreas, a PhD student in my lab, developed it. And basically what he was thinking about is like, what if we create a periodic line pattern? Nothing new. This is like typically used in structural illumination microscopy, right? So what you do, you focus on the back focal plane 2 point, and then, if you want a very fine periodicity, you try to space as much as possible these dots. But if you look at that sideway, so axially, you will see that look like tubes. So you don't have modulation axially. So what he thought is like, what if we do that? Like basically, we move this point on the side. Then you can create this oblique plane. And then you can do the same. So you see that you move the point on the side, right? So they interferes, and now they have this oblique illumination looking a sideway. So you can do that, and then you create another one that is oblique, but in the other direction. And these two, they don't see each other, they don't interfere with each other because they have different polarization. So when you put them on top of each other, they look like a honeycomb, like this. And this start to be interesting, because then if you have a fluorophore here, it start to be depleted also above and below the focal plane. So you start to have 3D confinement. And you can shine more and more light, and then switch more and more protein in the off state. basically imagine you have your sample over SFP, so reversible switchable protein. So the one that are here, they see the blue light, so they go in the off state. So you will only read the fluorescence coming from this tiny volume that now are confined not only in the XY, but also in XZ, creating like this 3D confined point spread function. In reality, we need another pattern to also have the other direction, the Y, and we still use microlenses, but it's more of a detail to get like, better contrast. And this is like the emission probability, right? So you have all this spot, because we are not doing spot by spot, but like many spots at the same time to save time. And these are a little bit like the data look like. So because we have microlenses, you see these dots. And then what you do, like conceptually, is exactly like you do in confocal microscopy. You integrate the signal and you reassign to a pixel. So you, in principle, if you want to be very robust, you don't apply any sort of the convolution or image processing. That doesn't mean that it cannot help, right? You can always do whatever you want with your image, add prior and get better contrast. But in principle, and I think this is the beauty of this method, that it can work with very simple integration. So in theory, without the convolution. That's why I'm saying it's very different than structural illumination. If you want, you can also apply on the very same data the algorithm that they use for nonlinear structural illumination, and probably you get some information here and there. But you also have to keep in mind that you need to know exactly where the pattern is, and you are putting some prior information on the type of point spread function you are using. So I always like to do it, because especially now we know the power of image analysis and what you can reach with that, but I think it's still very good to have the raw data as validation. And these are like some data that Francesca recorded. These are a cell that is expressing actually endogenously. So with CRISPR, we knock in the ResGFP with vimentin, and then you can see the Mona Lisa version. And for Mona Lisa, I mean this parallelized way of doing resolved. And then you see two things: that we are better in sectioning because we use the microlenses, but also that we have finer resolution. So you see that the filaments are thinner. You can see details that were hidden in the conventional microscope. You can decide to look at the parameter that you like the most. Sometimes some people like to quantify resolution to see the confinement that you can reach. Some other to look at the sort of relay criteria to see the distance, the minimal distance that you can distinguish still to spot. Both are meaningful. So here are a few numbers for both. Of course, you can confine up to 40, 50 nanometers, but then when they come together, it's more 70, 80 nanometers. And that you can actually improve with sort of the convolution, because then it helps you to sort of deal with the noise somehow. And here some validation also in origami. We convinced finally some friends to put their protein attached to two sides of the origami. And that was interesting to answer the question, how many protein do we need to reach a certain resolution? And then we could see that, we could go down up to 4 or 5 protein per site. And then these are sort of the nice result that you can get. For the one of you that are more into biology, so here we have labeled Homer, which is a postsynaptic densities in neurons. And we have applied our 3D imaging method. And what you see color-coded are the different axial position. And that's nice because you see, like, for example, here, green and violet were 2 points facing from each other, that in the projection image, you would say, oh, these are two synaptic site, but then you see that are basically first floor and last floor, so they're not actually so close to each other. And you have a lot of those artifacts if you live with projection or ignoring completely the axial resolution. So you have to think about that. And just to give you a number, like you can get hundreds of synapses in just a few seconds of recording. So in terms of speed and parallelization, we are actually entering an interesting regime. So just a reminder, so how do we do? So we have multiple spots that are confined, and we can look at the cell in 3D. And again, this is what I was stressing before. Yeah, we are moving from projection to having a more 3D information. And that to stress that, as a microscopy, I've been falling from the same things, like of working with projection, ignoring the axial resolution. But most of the time, because in many systems, the axial resolution is always worst, but then you have to deal with the fact that mistakes can be made. Maybe not as big as this one, where you can, mistake a hand for a wolf, or the other way around. But yes, this mistake can be done. And this, again, another argument where if you only work with projection, for example, in this case, what we were doing, we're taking a neuron and then induce a lot of activity, we call it long-term potentiation, and then checking what happened to these postsynaptic densities, if they increase or decrease. And sometimes if we would only look at the XY part, you wouldn't see a significant increase. But then if you look at the whole volume, you actually see that there is a big rearrangement on the morphology of the spine. So it's sort of really exciting now to move all this quantification and study that for historically has been done only in 2D to a more 3D system. And neurons are intrinsically 3D because are networks of cells. So it's important to have also this view. And here you're seeing, again, another volumes of a dendritic part. So it's a part of the neurons, and you're looking at spine head. And some colleagues like to look at the shape of the spine head. So here you can see these loops that you can only see sort of with axial resolution. So you can then pinpoint the morphology and then say it's a cup shape or other type of shape of the spine with much higher precision. And again, this recording takes only a few seconds. So that's also something to keep in mind for your throughput. And a lot of this study has been done, with tomography for a very long time. So now we can access that with light microscopy. It's quite exciting. And these are like some of the changes that you have when the neuron get crazy and start having activity. Then you get the spine that get bigger, actin get recruited, for example, and you can start studying also all these dynamic changes because these are recording done in a living cell. For the one of you that are more into metabolism, maybe you want to know the morphology of mitochondria and so on in 3D. So mitochondria are also a bit difficult because they move. So it's good to be faster. So it's one of the samples that benefit from this type of imaging. I guess you will hear more about mitochondria from Michelle. And here is just a view of how mitochondria look like with our system. So you can pinpoint the outer membrane. And what we are actually excited about is like to study their morphology, especially of these tiny vesicles that are budding out from them. So you can go basically in very crowded 3D parts, like the perinuclear mitochondria, and then start to look at them. And then you start realizing that some of them are actually not just the tubules, but they start protruding this tiny vesicle that can be a symptom of, getting rid of protein because the mitochondria is suffering, or the mitochondria want to give biogenesis to other organelles like peroxisome, and does this kind of thing. So that's what we are focusing on. And again, this can be done live. So those are all live cells. So you can study, for example, you can also perturb the system and see how they behave. You can say, hey, mitochondria, stop breathing. And then I'm going to check how you react. And then you see that at the beginning, they start this fission. So they develop in like they divide sort of, and then they start forming this fancy shape that I think physically has a lot of meaning. So where they start to make the membrane fuse, and then you get this sort of horseshoes type of shapes, and then eventually they start budding out also this tiny protein that probably have a lot of oxidated broken protein that they want to get rid of. So this is type of study you can do. And here again, so just since I showed that at the beginning and I was like, oh, we are motivated to get faster. Now, basically with the Mona Lisa, you can get this type of image, but at the same time. So this is like the exciting part. Let me see, how am I with time? Because now there are two other stories, so we can decide to go for one of the two. So Lionel, how long I have?
Can you hear me?
I can hear you now.
You have another 5 minutes.
Only 5, okay. Good. So then I move, because I know that Lionel is excited about polarization. So I leave you the event trigger part, and I move to this, which is pretty exciting. And it's completely different. So now you saw, like, you learn how to do volumetric imaging with photo switching, but now you're gonna learn how to measure rotational dynamics with photo switching. And say, why do we need that? Well, how do we measure binding, for example? For a very long time, and Lionel not that well, because there was a very big school that he has been involved with that has been doing like fluorescence anisotropy to measure how basically the dipole orientation of the molecule relax over time, and then with that infer something about the binding or not. So typically, like what you have fluorescent molecule in solution, you excite that with polarized light, and then the one that are aligned with the polarization of the light that has higher possibility to get excited, and then you analyze their emission with a polarizer in detection, and then you see basically how the photon change over time. And with that, you can get very interesting information, because you can get basically the rotational diffusivity, which tells you if something changed in the hydrodynamic radius. So if two molecules bind, the hydrodynamic radius get bigger, and you see you're super sensitive, the power of 3, then you will have a change in this rotational diffusion. Now, this is fantastic. So why we are not all using that to solve major biological questions in cells? The reason is that most of the protein in human are big, are like more than 30 kilodalton, or at least a huge portion are bigger. And then what means that if you do the same game, you don't see anything, because the molecule is just too big to rotate in this time. And so then we wanted to tag this issue. And that's where we come with this, our method that we name STARS, that stands for selective time resolved anisotropy with switchable state. And the idea here is to, is to get your molecule, your GFP, rigidly attached to a protein of interest, here in yellow. Then what you do, you shine light, the activation light that is polarized. What that means? That only this protein that I highlighted here will change conformation and will become visible. All the rest not. So you start having here a photo selection. And then, since you know how they were oriented, you can follow them over time and get your nice curve, still looking at the polarization in the two channels. But now you see that the kinetics is not anymore dictated by fluorescence, that you know the fluorescence lifetime is 1.5 nanoseconds. Now it's dictated by switching kinetics. And the switching kinetics can be tuned by light, can be several microseconds, even milliseconds. So that means you are not anymore restricted by the size of the molecule. If a molecule takes 100 microseconds to rotate, fine, then you shine the light in order to record their observable in longer time. So that's the key point. And of course, we did with photo switching because it's very light efficient, and we were happy that it works. In the past, and Lionel Mentor has been doing that also with the triplet state, which is also very interesting. Typically, for live cell is a little bit more problematic because it's more sensitive to oxygen, but you can find systems where it actually can work. But whatever you find along dark state, you can apply the similar principle. It's a little bit like super resolution. It can be simulated emission, it can be cis-trans isomerization, it can be triplet state population. And this was the wow moment for us when we realized that it's possible to imprint a photo selection during on-switching, because now it sounds, that happened like that, we take it for granted, but we didn't know, because there is a conformational change and the molecule can just screw up and have a completely different dipole. And it did change a little bit, but still not enough to let us lose completely the information. So, and this is Andrea that measured that. And then what we did here, we had the protein in the surface of beads of known size. And then we started measuring this anisotropy curve. And just by fitting this curve with non-exponential, we could actually find out the diameter of the beads. And then you see that it's quite interesting that you start measuring things that are, very close to their own size. So by looking in a time-resolved way about how this molecule, how this particle rotate, you can find out the size and then eventually their binding. And this is what we did, for example, with virus. We expressed that in HIV-like virus, and then we could see that in the immature virus, the protein are much less mobile than in the mature one, where it's cleaved away. So on the single particle level, we could say, oh, the virus is mature or immature. And we couldn't do that by super resolution because they look very similar. You see these two distributions are, yes, slightly different, but still very highly overlapping. So we couldn't have pinpoint on the single viral level. But then you can still get this super resolution image, analyze the photon in a very different way and see how molecules rotate in the microseconds range, and then find out if the virus is mature or immature. Okay, so that was a little bit fast, but just a teaser that, you know, you can do a lot of things with photo switching, not only imaging, and we are actually very excited to get more into spectroscopy in situ in a way, or combining different information. Yes, so I'm done. I'm looking forward for your question.